Welcome you all to my channel Omni Bio Entrance, and today we will be discussing IIT JAM Biotechnology question paper. I will be discussing only the biology part. This exam was conducted in the year 2013. If you are new to my channel, please do like, share and subscribe. I have uploaded still older papers of IIT JAM Biotechnology. You can refer to my channel playlist for the same. So coming to the first question. Continuous culture techniques at steady state, that is in chemostat, is useful in isolating bacteria from a mixed culture. Yes, that's correct. It exploits the difference in there. So continuous culture techniques, the growth rate of the microorganism is limited by the availability of nutrient, and this allows for the selection and isolation of different microorganisms with different growth rate. For example, the faster growing bacteria will outcompete the slower growing ones, enabling the isolation of the faster growing bacteria. So it exploits the difference in their specific growth rates. Answer is option B. The next one. Biofilms observed in the lungs of cystic fibrosis patient is composed of. So biofilm in cystic fibrosis patient, it is composed of polysaccharides such as alginate and Pseudomonas aeruginosa extracellular polysaccharides. These polysaccharides allow the bacteria to escape from the host immune system as well as become resistant to antibiotics. So the answer here is option A, that is polysaccharides. Next one. Match the pathogens listed in column 1 with the disease listed in column 2. So rotavirus. Rotavirus is for diarrhea, P2. So C and D can be omitted, it is P5 there. So Leishmania causes Kala Azar, Q3. So answer is option B. Salmonella typhimurium, it causes food poisoning, R1. And Epstein-Barr virus, it causes glandular fever. So the answer here is option B, P2, Q3, R1, S. Then next one. The time required for an E. coli to divide is less than the time required to replicate its chromosome. This is because of an abundance of enzyme required for replication. No. It linearizes the chromosome and replicates from both the ends. That's correct. It is bidirectional replication. Theta form of replication in E. coli. So the answer here is option B. Then next one. A bacterial population contains a mixture of wild type and leucine auxotrophs. From this mixture, the leucine auxotrophs can be enriched by growing the mixture in minimal medium supplemented with leucine and penicillin. Penicillin enrichment, it is a parallel technique available for auxotroph selection, and in this the penicillin is added to the liquid medium. The prototrophic cells, they are killed because they will divide in the liquid medium, which is minimal medium, liquid minimal medium. So in the minimal medium, the prototrophs will divide because of penicillin. They will be killed. But auxotrophs, they will survive because they are unable to divide there, and because there is no supplementation of leucine. Now what you do is you wash the cells and then plate the washed cells on a minimal medium with leucine. So the answer here is option C, that is leucine and penicillin. Then next one. Gram-positive bacteria are more rigid than gram-negative bacteria because of the presence of multiple layers of peptidoglycan. So gram-positive bacteria have thicker peptidoglycan layer composed of 40 layers, which make the cell wall more rigid, whereas gram-negative bacteria have peptidoglycan which is thinner and is made of 1 to 2 layers. Then next one. Helicobacter pylori can survive in the highly acidic environment of human stomach because it creates an alkaline microenvironment around itself by urease action. That's correct. Helicobacter pylori, it produces urease which catalyzes the hydrolysis of urea into carbonic acid and ammonia. The ammonia increases the pH around the bacterium, making it alkaline, and it provides a protective environment that allows it to survive in the acidic stomach. So the answer is option A. Then next one. The waxy surface of dental plaques does not allow oxygen to diffuse rapidly. A sample of dental plaque was inoculated into a rich medium and incubated at 37 degrees Celsius with vigorous shaking. However, there was no bacterial growth. The absence of growth was attributed to the following reasons. These bacteria are obligate anaerobes. These bacteria require highly alkaline medium for growth. These bacteria require a very hard surface such as that of teeth to grow. Which of these reasons are likely to be correct? So since there was no bacterial growth upon vigorous shaking, that is you supply more oxygen to it through vigorous shaking and you inoculate into a rich medium that is filled with nutrients, the bacteria didn't grow. That means these bacteria are obligate anaerobes. So P is correct. These bacteria require highly alkaline medium for growth. That is not correct. These bacteria require a very hard surface such as that of teeth to grow. It is also correct. Because in the medium that you provide, it didn't have any hard surface in it. So that also could be and it was oxygenated also. So because of this reason, the obligate anaerobe didn't grow. So P and R are correct. So answer here is option D. Then next one. Cellulases and hemicellulases are in great demand nowadays for the preparation of second generation bioethanol. Because cellulases and hemicellulases, they are enzymes and they break down the plant cell walls releasing sugars that are converted into bioethanol, which are second generation fuels. Then next one. Marine algae are very important due to their potential industrial significance as single cell oil. These can be used for which of the following? Biofuel production, recombinant insulin production, atmospheric carbon dioxide sequestration, antibody production, polyunsaturated fatty acid production. So marine algae can be used in the production of biofuels, then atmospheric carbon dioxide sequestration and polyunsaturated fatty acid production. But they cannot be used for recombinant insulin production where you use E. coli. Then antibody production also they cannot be used. For monoclonal antibodies and all you cannot use microalgae. So the answer here is PRT. Answer is option A. Then match the enzymes in column 1 with their commercial application in column 2. Alkaline protease. Alkaline protease is detergent formulation. Proteases are used in detergents. So P4. So B and C can be omitted. We will see A and D. Asparginase. Asparginase is treatment of lymphocytic leukemia. It is used. Asparginase is used there. Q6. So A is the answer. Immobilized Q5 is given in option D. So it is not correct. Then immobilized aminoacylase. Immobilized aminoacylase is used in resolution of D and L amino acids and drugs. R5. Lipase is used in production of biodiesel and immobilized glucose oxidase is used as a biosensor. It is used in biosensor. So the answer here is option A. Then next one. A monoclonal antibody against a specific cell surface receptor was generated. This was expected to block the receptor function. However, it led to the activation instead of blocking. This could be due to. So the monoclonal antibody, it was generated against a specific cell receptor to block the function of the receptor. But what happened was it got the receptor activated And this could be due to cross-linking of the receptors by monoclonal antibody resulting in receptor activation. That is the answer. Answer is option A. Then PFU polymerase has got proofreading activity. It is a polymerase similar to Taq polymerase, and it can be active even at higher temperatures, just like the Taq polymerase. But Taq polymerase doesn't have proofreading activity, but PFU has proofreading activity. Then next one. Match the biotech products listed in column 1 with applications in column 2. Polyhydroxy alkanates, so PHA, they are in bioplastics, P4. So A and C can be omitted. We will see B and D. Biosurfactants or bioemulsifiers, they are microbial enhanced oil recovery. 6-aminopenicillinic acid. It is used in precursor for beta-lactam antibiotics. That is like ampicillin, amoxicillin, all are prepared using 6-aminopenicillinic acid. Docosahexaenoic acid. It is a nutraceutical or functional food. And dextran, it is used in size exclusion chromatography. So answer is option B, P4Q6R5S2T1. Then next one. Match the material listed in column 1 with the techniques, purposes listed in column 2. Phenyl agarose. Phenyl agarose, it is used in hydrophobic interaction chromatography, P3. Answer is option C. P3 is there only in option C. NINTA matrix. It is in affinity chromatography matrix for purifying recombinant proteins carrying a his tag. So Q is 5. Affinity chromatography. TEMED is used in gel electrophoresis of proteins, R4. You prepare the polyacrylamide gel by using TEMED. Then silicone oil. Silicone oil is an anti-foaming agent. So answer is option C, that is P3, Q5, R4 and S1. Then next one. Determine the correctness or otherwise of the following assertion. HeLa cells derived from cervical cancer tissue and fibroblast from a normal human male cannot divide indefinitely in cell culture. Cells undergo senescence, that is aging, even when grown in cell culture. That is correct. That is the reason for A. But instead of cervical cancer, it is not found in human males, it is found in females. So it might be a printing mistake, but anyway, both A and R are true, and R is the correct reason for A. That is the correct answer. Okay. A human gene was PCR amplified from a cDNA library, cloned into an expression vector and transformed into E. coli. However, the expression of this gene was poor. This is because eukaryotic mRNAs, they have different structural features and codon usage compared to bacterial mRNAs, which can lead to poor translation efficiency in E. coli. So the answer here is the incompatibility of the mRNA with E. coli ribosomes. Answer is option C. Then next one. A student wanted to clone a gene in a vector, that is 1.6 kb size, in a vector of size 5 kb. The vector has sites for restriction enzyme RE1, RE2, RE3 in its multiple cloning site. The site for RE2 is located in between the sites for RE1 and RE3. The student carried out the following steps for cloning. Cut the gene G with restriction enzymes RE1 and RE3. Okay. Cut the vector V with restriction enzymes RE1 and RE3. Ligate G and V from steps 1 and 2 transformed into E. coli and isolate plasmid L from the transformant. So plasmid L is having your gene of insert in it. The plasmid L was cut with RE2 which gave a band of size 6.6 kb. That is 5 plus 1.6 kb will be 6.6 kb. So it got inserted there. Your gene of insert got inserted into the plasmid L. The following three reasons were given to explain this observation. Plasmid L is the same as vector V. No. Plasmid L is having your gene of interest whereas the vector V doesn't have your gene of interest. So P is not correct. Gene G has site for restriction enzyme 2. That's correct. That is why you got a size of 6.6 kb upon digestion with restriction enzyme 2 for plasmid L. So the gene G, that is your gene of interest, it is having a site for RE2, restriction enzyme 2. Then RE1 and RE2 are isoschisomers. So isoschisomers, they are restriction enzymes that have the same recognition sequence and cleave the DNA at the same positions. But they need isoschisomers, they need different reaction conditions like pH, ionic strength and even temperature. But here they have not mentioned anything regarding that restriction enzyme 1 and 2 require different conditions, different reaction conditions they have not mentioned. So they cannot be isoschisomers. So R is also not correct. So answer here is option D, that is Q alone, neither P nor QR. Then next one. The following is a schematic of lac operon. Which of the following statement is correct? Now before coming to the options, let me tell you that cis acting is an element. An element is considered to be cis acting if it is has effect on the same DNA strand on which it is located. That is cis acting. And trans acting means it will be located on one DNA strand but it will have its action on another DNA strand. That is trans acting. Now lac-I being part of an operon. Lac-I is not a part of the operon and it is not a structural gene also. So A is not correct. P being furthest from lac-A is trans acting. P is not trans acting, it is cis acting. It is present, lac-A is present on the same DNA strand and it is cis acting. So B is also not correct. Lac is said is the structural gene and it codes for an enzyme. That is correct. It codes for beta lactosidase. Lac-I is cis acting. Lac-I is trans acting. Lac-I is not present on the is not a part of the operon and it is found near to the lac operon but it is not a part of the operon. So it is present on a different DNA strand. So it is trans acting. It is not cis acting. So D is also not correct. Then next one. Mammalian cells in the S phase can be monitored by incorporating which of the following in the cell culture medium. So S phase means they are in the DNA replication phase. So if you add H3 thymidine, you can monitor them. Then next one. The following experiment were performed with a double-stranded DNA sample S of the size 1 kb. The two strands were separated. Step 1. The separated strands were annealed. Okay. Treated the double-stranded DNA from step 2 with 3' 5' exonuclease. From agarose gel electrophoresis, it was found that the DNA from step 3 is less than 1 kb. The most probable reason for not getting 1 kb DNA in step 3 is that the DNA in sample S is palindromic. No harm, you will get 1 kb. Has sequence repeats, still you will get 1 kb. Has sequence inversion, then also you will get 1 kb. Has strands, each of which has self-complementary sequence. That is correct. Self-complementary, it refers that the sequence of DNA may fold back on itself, creating a double strand-like structure. And this decreases the size of the sample. Then next one. A batch of students studied the effect of changing either phospholipid composition or temperature on the transport of solutes across liposomal membranes. From these studies, the students arrived the following conclusion. Increase in the saturation of hydrocarbon chain lead to an increase in the rate of diffusion of oxygen. It will not increase the rate of diffusion of oxygen, but it will decrease the rate of diffusion of oxygen across the lysosomal membrane. So P is not correct, because saturated hydrocarbon chains, they are more rigid and they are tightly packed when compared to the unsaturated hydrocarbon chains. So when you increase the saturation, it will lead to less fluidity of the membrane and less permeability to small molecules such as oxygen. So increasing saturation will lead to decrease in the rate of diffusion of oxygen. So P is incorrect. Then Q. Decreasing the temperature to 5 degrees lead to a decrease in the rate of potassium transport by valinomycin, a carrier ionophore. So valinomycin, it is a carrier ionophore that facilitate the transport of potassium across the membrane. So in this process, the valinomycin and potassium complexes cross the membrane, and this crossing, it is influenced by temperature, and lowering the temperature, there will be decrease in the kinetic energy of the molecule and reduces the fluidity of the membrane and slow down the diffusion process. So Q is correct. So decreasing the temperature to 5 degrees lead to decrease in the rate of potassium transport by valinomycin. Q is correct. Then next one. Increase in the hydrocarbon chain length did not have any effect on the transport mediated by gramicidine, a channel ionophore. So gramicidine, it is an ionophore that forms channels in the membrane allowing ion to pass through. And they have mentioned they have increased the hydrocarbon chain length, but hydrocarbon chain length does not affect the transport of ions mediated by gramicidine. Because gramicidine mediated transport is driven primarily by electrochemical gradient across the membrane. So increasing the hydrocarbon chain length will not have any effect on the transport mediated by gramicidine. So R is also correct. The question is which of the conclusion is not correct. So P only is incorrect. Answer is option C. Then next one. Circular DNA of bacterium was isolated to replicate once. In this experiment, some of the nucleotides were fluorescently labeled. The primase was able to accept the labeled as well as unlabeled nucleotides. However, the DNA polymerase could use only the unlabeled nucleotides. The fluorescent label is found in. So the primase can accept both labeled and unlabeled nucleotides. The primer will be labeled. Okay, the primer will be labeled. DNA polymerase can use only unlabeled nucleotides. So the newly synthesized strand, it will not be labeled, but the primer region will be labeled. The template strand means it is the original circular DNA which is not affected by the labeling. So during replication, the primer is extended by the DNA polymerase, creating a new strand. And the primer is labeled, I already told you. So the new strand will also contain the labeled nucleotides. So the answer here is option B, only the newly made strand. Then next one. Consider the following metabolic pathway found in bacteria. S2I1 I2 to P, catalyzed by enzyme E1, E2 and E3. E1, E2, E3 are enzymes which catalyzes different steps of the pathway as indicated above. There are three classes of mutants. Class 1 has defective E1, hence requires L1 in the medium to grow. Class 2 has defective E2, hence requires L2 in the medium to grow. Class 3 has defective E3, hence requires P in the medium to grow. Wild type grows in minimal medium. Okay. We mix class 1, class 2, class 3 mutants. Allow this mixture to grow in minimal medium. Suppose we observe growth because of revertance. So some of them have got reverted to the wild type. Then the reversion has to be in. Now suppose if class 1 got reverted, then there will be gain of function of E1 and they can grow in minimal medium. Now suppose class 2 mutants, they require L2 for growth. Now if they got reverted, there will be gain of function of E2 and they can grow in minimal medium. Now class 3 mutants, they require P to grow. So if they revert, they can grow in the minimal medium and produce P. So the answer here is any of the three classes. Since the reversion can occur in any of the three classes, the growth observed in the mixture could be due to the reversion of any or one or more of the three classes. So the answer here is option D. Then next one. The stain DAPI is used for total cell count because it reacts with DNA. DAPI, it is a fluorescent dye that specifically binds to double-stranded DNA and it is useful for visualizing cells with their nuclei. Then next one. Which one of the following statement about cytoskeletal protein is not true? They give shape to the cell. That's correct. They help in cell division. That's also correct. They help in cell motility. That's also correct. They are non-essential proteins. No, they are essential proteins. So answer here is option B. Then next one. DNA was isolated from 3 samples of the normal human cell. In each of the samples, cells were at different stages of cell division. Cells were in G2 phase, sample 2 cells were in anaphase, sample 3 cells were in telophase. DNA isolated from these samples confirms the Chargaff's rule. Which of these samples confirm Chargaff's rule? So Chargaff rule, it states that DNA, in a double-stranded DNA, the amount of A is equal to the amount of T and G is equal to the amount of C. So this rule applies to the DNA molecule as a whole and regardless of whichever stage of cell division it is. Now cells in the G2 phase, the DNA content is doubled, but the ratio of AT and GC, A is to T and G is to C will remain the same. Cells in the anaphase, the DNA is condensed and separated, but the ratio of A is to T and G is to C remain the same. And in telophase, they are fully condensed and the ratio of A is to T and G is to C remain the same. So all three will confirm Chargaff's rule. So answer is option D, sample 1, 2 and 3. Then next one. Which of the following features are shared by mitochondria and chloroplast? The energy transducing membrane is invaginated. Yes, in both chloroplast and mitochondria you can see invaginations. Stroma of chloroplast and matrix of mitochondria are alkaline. That's also correct. Their number per cell is variable. That is also correct. So the answer here is option D, P, Q and R. The next one. Cilia and flagella are locomotor appendages found on some cells. A flagellum has an undulatory motion. A cilium has back and forth motion. The following statements relate to the direction of movement of a cell to the axis of flagellum or cilium that drives its motion. P. The cell moves along a direction perpendicular to the axis of the flagellum. That's wrong. The cell moves along a direction parallel to the axis of the flagellum. It is not perpendicular. Q. The cell moves along a direction parallel to the axis of the cilium. No, the cell moves in a direction perpendicular to the axis of the cilium. So both the statements are incorrect and your answer is option C, neither P nor Q. Then listed below are some classes of enzymes. Which of the above are associated with post-translational modification of proteins? Exoproteases. Proteases are protein degrading, so they are naturally not going to be involved in post-translational modification of proteins. Glycosidases, no. Glycosyl transferases, yes. R and lipid transferases, yes. So D, R and S is the answer. Cell A is in S phase of cell division. Cell B is G1 phase of cell division. If cell A and cell B were to be fused, then the nucleus of the cell B in the heterokaryon will be. So DNA replication is expected to be induced in G1 cells fused with S phase cells due to the presence of S phase factors that initiate DNA replication. So the answer here is it enters. That is, when S phase cell is fused with a G1 phase, cell in G1 phase of cell division, the fused nucleus of the cell B in the heterokaryon will enter the S phase. So that's it. Thank you for watching my channel. Please do like, share and subscribe.